All right, continuing with our discussion of carbohydrates. We're now going to turn to disaccharides. And disaccharides consist of two monosaccharides, which are simple sugars, polyhydroxy, aldehydes, and ketones, usually from 3 to 8 carbons, with 4, 5, and 6 carbons being the most common. When two monosaccharides are linked together, we have a disaccharide. There are 3 naturally occurring glycosidic linkages. There's a 1 to 4 prime link, where the anomeric carbon is bonded to the oxygen on C4 of the second sugar. There's a 1 to 6 link, where the anomeric carbon is bonded to the oxygen on C6 of the second sugar. And there is a 1 to 1 link, where both anomeric carbons are bonded to each other through an oxygen. Okay, so here we have beta-D-glucopyranose, and okay, here we have a glycosidic bond, and if that glycosidic bond is to another sugar, then we have a disaccharide. This is a 1 to 4 prime linkage. Carbon number 1 of the sugar on the left is bonded to carbon number 4 of the sugar on the right. Okay, and if the first glucose has the anomeric carbon in the beta configuration, and we have a a 1 to 4 prime beta glycosidic linkage. These are both glucose molecules. This is a compound called cellubiose. Okay, and this is the long name for cellubiose. But the important thing here is the configuration at the anomeric carbon. This is beta. Okay, this portion of the molecule does equilibrate between alpha and beta, but this part is fixed. Okay, this is an alpha 1 to 4 prime glycosidic linkage, and this is a disaccharide known as maltose. Okay, so again, this configuration is fixed, but then this ring can open and close. Cellubiose is a component of a larger polysaccharide called cellulose, where thousands of these units repeat themselves with all beta 1 to 4 prime glycosidic linkages. And if we repeat this unit thousands of times, then we have starch, or we have at least the amylose portion of starch. Okay, this is maltose. The longer name is 4-O-alpha-D-glucopyranosyl D-glucopyranose. Maltose is malt sugar. And if you're familiar with malted milkshakes or malted milk candy, okay, like Whoppers or Robin's eggs, okay, that flavor comes from this disaccharide called maltose. Right, this is lactose or milk sugar. The difference here between lactose and cellulose is that this is galactose rather than glucose on the left. Lactose is the major carbohydrate in milk, and the difference between galactose and glucose is that at the C4 position, the OH is axial in galactose rather than equatorial. Okay, so we form a beta galactosidic linkage. And this lactose cannot be digested. It's first got to be broken down into galactose and glucose. And this requires an enzyme called lactase. Okay, here we have a disaccharide called gentiobiose, and this is a 1 to 6 prime beta glycosidic linkage. This is rare for disaccharides, but it's commonly seen as a branch point in carbohydrates. Two examples are the amylopectin portion of starch, okay, which contains branch units, and glycogen, which is the storage form of glucose in animals. Glycogen is very highly branched. It contains a lot of these 1 to 6 prime linkages. Okay, here is sucrose. Okay, ordinary table sugar. And it consists of a glucose and a fructose unit. And it is a disaccharide where the two monosaccharides are joined by their anomeric carbons. So this is a 1 to 1 prime linkage. Sucrose is an example of a non-reducing sugar. Okay, it will not form osazones. It will not react with Tollens or Benedict's reagent. Okay, the stereochemistry here is important for, in the case of the glucose, the anomer has to be alpha, and in the case of the fructose, it has to be beta. All right, if we repeat the cellulobiose disaccharide many thousands of times, we have cellulose. Cellulose is the most abundant organic material in nature. It forms the cell walls of plants. Paper is largely cellulose. Cotton is mostly cellulose. It's synthesized by plants as a structural material to support the weight of the plant. Cellulose is not digestible by humans. It is digestible by certain ruminant animals, okay, such as cows, sheep, camels, goats. But even then, the digestion takes place as a result of a symbiotic relationship with a microorganism, which produces the necessary enzyme to break down cellulose into glucose. Humans and many other animals use an enzyme called amylase to break down starch. The amylase enzyme recognizes the all-alpha configurations in starch and is able to break down starch into glucose. The other organism that can digest cellulose is another example is wood-rotting fungi. Okay, so here's amylose, and Amylose actually has a different three-dimensional shape. It's like a coiled spring. Okay, here's amylopectin. This is another part of starch, which has several of these branch points. Okay, next we'll turn to nucleic acids. Nucleic acids are polymers of ribofuranoside rings that are linked by phosphate ester groups, or also called phosphodiester groups. Okay, each of the ribose units is bonded to a base. There are 4 bases in DNA and 4 in RNA. Okay, if the sugar is ribose, then this polymer is ribonucleic acid, or RNA. And if it's 2' deoxyribose, then it's DNA. For DNA, at this position, we would have an H instead of an OH. Okay, so here's an RNA polymer. Okay, at one terminus, we have the 3' end, and at the other terminus, we have a 5' end. So it's a 3,5 to 5' phosphodiester linkage. And at the 1 position, we have the various bases that are bonded. All right, so these are the 4 nucleosides from the bases for RNA. We have cytidine, uridine, adenosine, and guanosine. Okay, which are derived from cytosine, uracil, adenosine, adenine, and guanine. Okay, so besides RNA, we have some other common ribonucleotides. The difference between a nucleoside and a nucleotide, these are nucleosides. Nucleotides will also have a phosphate group. Okay, so this is called cytidine monophosphate, or CMP. Okay, by forming an ester between phosphoric acid and this alcohol, we produce our phosphodiester linkages. These are the bases of DNA. The difference between DNA and RNA is that instead of uridine, we'll have thymine. Uridine does not have this methyl group here, but the other 3 bases are in common. Okay, so in the structure of DNA, we have the beta D2-deoxyribofuranose for the sugar, and we have 4 heterocyclic bases. These are linked by phosphodiester groups to form the primary structure. And then we have base pairing between strands. So guanine pairs with cytosine, adenine pairs with thymine. Okay, so the strands of DNA are anti-parallel. That is, they are in the reverse sequence of each other. So A pairs with T, G pairs with C, C pairs with G. Okay, so the 5' and the 3' are at opposite ends for the two strands. Okay, and when this base pairing takes place, this results in a double helix shape for DNA. And this is a right-hand double helix. This double helix structure was described by Watson and Crick in 1953, and they were later awarded the Nobel Prize for this discovery. Okay, this is a simplification, but when DNA replicates, the two strands unwind, and and then two new double helices are produced by anti-parallel base pairing. Okay, some other nucleotides include adenosine monophosphate, or AMP, which is a regulatory hormone, nicotinamide adenine dinucleotide, or NAD, that's a coenzyme, and adenine triphosphate, an energy source. Okay, so that is all for that chapter.